So now our next speaker is Shankar from Beckman Coulter, and he's going to talk about DuraClone antibody panel technology and how it can be used for the biomarker detection. So Shankar, it's over to you.
So today, hello, good morning, good afternoon almost. So I have gone through all the presentations so far, and I think most of them have been about different technologies and how the need is to standardize and have a very clear measure of what you're trying to identify, rather than just producing a lot of data which is not very consistent. I think that's what we are also working on, especially in Beckman Coulter. Though we are not actively into the aging or the longevity field, we are mostly studying the immune system, and that's what Beckman Coulter has been doing for the last uh, 50 years, I would say. That we mainly have this technology of flow cytometry, where we are able to analyze huge number of particle data, that is anything which is in a cell or any microbe, any bacteria, any particle which is there can be measured in large quantities and can be used for very good system biological studies. And flow cytometry is predominantly used for the immune system, because the immune system is very well characterized with the antibodies on the surface of the different immune cells, and flow cytometry can be a very powerful technique to identify these kind of cells. And it has got a huge number of applications. So you have applications in basically everything: immune profiling, drug action, protein analysis, biomarker discovery, huge amount of applications. And I think in the last 50 years, flow cytometry has been extensively applied for most of the system biological systems. So there's a lot of data being produced, good quality data being produced, and you have a very good snapshot of the immune system at any state. So what are we doing in Beckman Coulter is that we want to make this whole business of flow cytometry easier for people. Flow cytometry is complicated because you need to have thousands of hundreds of antibodies with you, need to mix them, you make mistakes in mixing them, you don't have good quality reagents, you need cold storage, you need to transport material, it's all very complex business. So flow cytometry in any institute or any place you go is a big thing. But what we want to do it is make it a very easy thing, easy for people to use. And the most important thing that we came up with is the dry technology, which is we are able to keep the antibodies in a dry state. It is not a live state, but in a dry state at room temperature. So you don't need any cold storage, you carry them with you at room temperature. And this whole technology was developed here in India, here in Bangalore. And we have our manufacturing facility also in Bangalore, and we can say proudly that we are completely discovered in India, invented in India, and make in India out here for this thing. And we export throughout the world. So about this. So what we have is very antibody panels, which we have already well characterized antibody panels, which can reveal different facets of the immune system. Because I think one of the most important surveillance mechanism, or the thing which actually controls the whole human body, is the immune system. Any perturbation in the immune system is going to have a profound effect from the top to the bottom. Every organ is going to be affected by it, and every disease or every microbe or any bacteria or virus has to first interact with the immune system. So to have a good understanding and an in-depth understanding of the immune system is pretty essential for any biological question, and especially the aging issues because of the changes in the immune system that occurs when the person ages. So we have a set of markers, as I put out there, a set of panels out there which identify, which look at different aspects of the immune system. One is immune monitoring. What are the different types of subsets of the cells, B cells, subsets, T cell subsets, granulocytes, all the basic subsets of the immune system. You can identify them and quantitate them. Then you have the immune function things. How are the immune cells functioning? Are they what's it called responding to stimulus in the same way? Is there a decrease in the response in the functional activity? Then you have rare events. How to identify rare cells in the immune system, and then also now coming down to the stem cells, because that is again a direct relationship to what, how good, how healthy is your stem cell population, because this is what is going to lead to how good your body is. Basically we are looking at the mesenchymal and hematopoietic stem cells, so you do need a healthy population of these two to have a prolonged immune system which is healthy and functioning. Why are we doing all these things is because we want to make flow cytometry easy and simple. Our flow cytometric tests are very easy to use. They are just 3 steps to do. These have been extensively used especially in conditions where normally you can't take antibodies. So in the field where you can go to the villages, you can go to small towns, you can collect blood samples, you can transport all your material back to the lab easily, so that you can do this analysis of the immune system. It is also used for disease identification. Of course, the primary application for all the flow cytometry is basically leukemia, lymphoma identification, blood cancers. But you can also do simple immunoprofiling of patients, of people, using this technology. It's easy to use, it reduces human error, there is no pipetting required, only blood needs to be pipetted into it. It is easy to transport. It is, and this is this is our technology that we have developed here. The dry unitized reagent assay, where you don't need any pipetting except putting the blood into it. It's a very essentially the moment we brought it out, everyone liked it, because this is ideal for clinical studies, because you have the same reagent in multiple sites. You don't need to have, normally what would happen is, you would suppose you have an antibody, you will have 5 batches of the antibody from the company. You have to send each batch to each of the multicentric site you have, and you will have variations in the batches. Here you have one batch of reagent sent to all the sites. You have stability, easy to use, all the data is very reproducible and comparable, and that is the hallmark, and that is the most important thing you require for clinical studies, that they have to be comparable, reproducible. And we have extensive number of publications where people have been using these kind of what we call panels for doing clinical studies, because this is I think very essential for this field. Yes, so what we don't only supply reagents, but we supply reagents, that is our bread and butter, but we also supply automation, because again that is another source of extreme amount of variation that you find in it, and it's also very laborious. So we also offer automation where it's true walk away. You put the blood samples and you just walk away, and the 24 hours the instrument will prepare its own samples. And you have the analyzers, which are also are here. Actually in IISc you have many of these analyzers which are being used for analysis. And we also have a lot of softwares which are for deep analysis. Especially I wanted to mention about this Cytobank software. This is for the meta-analysis of the immune system. Basically you can combine an enormous amount of data to study different cell populations and how they are altering the conditions. Yeah, so basically what we are talking about is a robust technology of how to study the immune system in a very standardized, in a very proper way, so that you get very reliable results. If you ask me, I don't have any data for the aging research, but we are working with the teams here in IISc, and we are contributing our reagents for it, so that we will be now generating data for how does the immune system alter during the aging process. And this, we just wanted to quickly show this this automation system, because I think it's pretty essential. Any person who has done flow cytometry will know that automation can be a big boost for them. It really helps the lab streamline its workflow. And I think this is the future for flow cytometry and immune studies. Thank you. Any questions? Yeah.
Although you said you guys have not done much work in aging,
Yeah.
but have you looked at neutrophil to lymphocyte ratio in another context,
Yeah.
and how does it differ in Indian population?
I don't have the data for that.
Okay, it will be interesting to look at
Absolutely.
it from aging perspective, because it's known to be a global marker for systemic inflammation, and that's the first thing that goes up. The second question is collaborations, and then I can talk about that later on with you.
So we have groups in IISc, especially Siddharth and Deepak group, who are actually using this thing. So they will be generating data. So we are actually want them to generate as much data and publish it, so that we can also say that our technology has been used. But we ourselves have not worked on this field.
Okay, and what's the timeline for the data, from data to results?
Ask him. How much time will it take for you to generate the data?
It's sample collection is a major problem.
So if it's not there, if we provide you the samples, how long we get the results?
It's not very long. It's about a few hours.
Few
Okay,
hours.
okay.
Yeah, it's a few hours of data. So it's the same day.
Yeah. The follow up on NLR is not a good marker for anything apart from everybody knows neutrophil
Yeah.
goes up.
Oh, it's basic marker at least
Basic
to
marker
begin with.
for clinicians. It's a very
Yeah,
good
yeah.
marker, but beyond that, I don't think.
In
lot of
preventive care,
India-
it is a great marker,
Yeah.
I think.
It's the first thing any infection, any inflammation,
Exactly.
neutrophils will go up. So
Exactly.
you'll know that's why complete blood counts are done. So beyond
Yeah.
that, I don't see a major change.
Yeah, okay.
But we do expect that they would generate very high quality data, and I think this data would be, I think, very useful for I think this is the first step in this field. I have not seen much publications with the study of immune system during aging. I think we are just waiting for them to get it done.
Thank you.
Yeah,
All right. So, any more questions?
yeah, here.
Great talk, and yeah, we've been using your product for running our samples. So I think an issue we face is the red blood cells that are present over there.
Yeah.
So we usually lyse them and then
Yeah.
run it over here. But then your DuraClone product states that you don't need to do it. But
Yeah.
still, there's a lot of noise that is generated. So is there any workaround around
So I mean, you need to remove the red blood cells. But I have also told that I will be talking to your people to see how we can actually. There are protocols where we can actually make that much easier. So that is the current technology where we need to lyse the red blood cells, remove them from it, and then go for it. But now we are coming up with new technologies where that might not be even required. So I think you will see it in the next 1 or 2 years, probably, technology where we have made great advances, where you will see that you can do much more with the data you're generating, and it's going to be much easier for you to generate the data.
the grid?
All right. So, if there is
Thank
no
you.
more questions, so let's thank Shankar for a wonderful talk.
Thank you.